Hello everybody. Welcome to our webinar Wednesday, now for the second time. I'm Monika Burber, Product Manager for Biochemicals and Instruments at Biozyme, with over 20 years of experience in molecular biology. We are proud that so many people, customers and curious scientific workers join our webinar today. Thank you. The aim of Biozyme webinar is to transport new and exciting information about technology and new products to all interested people. Today, we will introduce riboPOOL for efficient and flexible ribosomal RNA depletion. I'm happy that today, Catherine Go from the company siTOOLs presents new data and interesting background information about the riboPOOLs. At the end of the session, there will be time for a discussion. Please type your question into the chat box whenever you like. Catherine, tell us more about the riboPOOLs.
Thank you, Monika, and hello, everyone. Thank you for joining us from wherever you are. I'd like to thank Biozyme, our German-Austrian distributor, for hosting this webinar today and giving us the opportunity to share more about riboPOOLs for ribosomal RNA depletion. So I'll just get started now. Just a short introduction about siTOOLs Biotech. We're a biotech based in Munich in the Martins-Ried biotech cluster. We were founded about 7 years ago as a spin-out of Intana Bioscience and the University of Regensburg. We're an all-scientist team, and we're really experts in producing and designing oligonucleotide-based reagents. And because our founders were RNA-I screeners, we have a very strong foundation and expertise also in RNA interference, which is a method of genetic disruption. And our products all share a common theme. So they're basically pools, complex pools of optimally designed oligos that are used to examine gene function. And we also offer services around our RNA-I expertise, which mainly focuses on screening projects and data expression analysis. So why pool oligos? We discovered the power of pooling, mainly with RNA interference, which we started out with. And we found that it also applied to the riboPOOLs or biotinylated probes. So what happens when we typically use a single oligo or even a low complexity pool of 3 to 4 oligos is that not only does one hit the target gene or transcript, you often see other transcripts being affected as well. So this leads to some side effects. It can lead to functional side effects in terms of RNA-I, or with biotinylated probes, it can lead to non-specific binding. And because of this, also, there's another effect, which is with having a single oligo, you only have one shot on target, so it binds only to a single site on the transcript. And if this site is inaccessible, this single oligo will not work. And we see this also happening with RNA-I, where siRNAs tend to be very variable and not all of them work. So this is the reason why we use a pool of oligos. And by a pool, I mean typically 30 or more oligos. With riboPOOLs, they're much more complex than 30, which is used for siPOOLs. And with more oligos in the pool, one would think, wouldn't you get more off-target effects? But the key thing to bear in mind really is the concentration that you're applying the pool at. And the total concentration of the pool will be much. And at this concentration, the individual oligos are present at a much lower amount than what you would typically expect. So the off-target effects that are specific to each oligo are much reduced. However, all the oligos in that pool would bind to the transcript on different sites. So you also get a very good coverage of the transcript and hence a better reproducibility of results. So more efficient binding or targeting, which is why we use complex pools. And we've published a paper with the siPOOLs, really going into detail on the function and how it works, which can be found on our website. But for today, we are focusing on the riboPOOLs, which I'll show you some data on. And we also have another product called rPOOLs, which are chemically quite similar to riboPOOLs, but are used more for RNA enrichment instead of depletion. So how we got started with riboPOOLs really began with an unusual model organism. This is a freshwater worm called the planarian. And what's amazing about this worm is its incredible capacity to regenerate. So if you can see here, when cut into pieces, each piece of the worm is really able to, over a couple of days, regenerate into the complete worm because of its high abundance of pluripotent stem cells. And we had a group from the University of Bayreuth who were studying this organism and came to us with a request. They wanted to study what genes were involved in this regenerative capacity. And they had a suspicion it could be small non-coding RNAs that were involved. So what they did was to knock down by RNA interference these two proteins, SMED-V2 and SMED-V3, which bind to these small non-coding RNAs to form a complex called the pi-RISC, which regulates gene expression. So they were knocking down these proteins and doing RNA sequencing to see what genes were up or down regulated. And for this, they required ribosomal RNA depletion to before they could do the RNA sequencing. So because there wasn't a tool at the time for planarians, they came to us with a request if we could make these custom oligo pools, since we were doing rPOOLs at the time. So why do we have to perform ribosomal RNA depletion? I think most of you are familiar that ribosomal RNA take up 80 to 90% of the total RNA for many species. And this therefore limits the sensitivity of detection of relevant RNAs that we like to study, which are messenger RNAs, for example, or other non-coding RNAs. So they have to be removed before RNA sequencing, otherwise it leads to very costly expenditure on reads that go to ribosomal RNAs. There are two current techniques currently to manage this problem. The first is the poly-A selection, which is mainly enriching for poly-A containing transcripts with DT oligos and isolating these transcripts for sequencing. Now, most people do this when they're interested in coding genes, so exons. However, this method does tend to miss out on non-polyadenylated RNAs, which can also be coding genes such as histones, as well as long non-coding RNAs or small non-coding RNAs. And also for bacteria or prokaryotic RNA, these are typically not so well polyadenylated, so this method is not well used in this field. With this method, we also lose information on immature transcripts or transposons or processed transcripts lacking a poly-A tail. And because it relies on intact poly-A transcript, it also doesn't perform very well for degraded RNA or formalin-fixed samples, and tends to have more reads focusing on the 3' end. So physical rRNA depletion, which is what the riboPOOLs are based on, involves DNA probes that are designed against ribosomal RNA, and these are linked to a biotin. So when using streptavidin-coated beads, this hybridized rRNA in the probes can be removed, leaving only the relevant RNAs for sequencing. And what's advantageous about this method is its ability to detect small and long polyadenylated RNAs, and also its better performance on degraded RNAs and FFPE samples. And it can also be used for prokaryotes without a 3' bias. However, one does have to go quite deep in terms of sequencing reads, simply because there's so much more information to capture. So the group from the University of Bayreuth, Dr. Klaus Kuhn, published a paper using the riboPOOLs, and they compared it with current poly-A libraries sequenced in the past. And what they found was that the riboPOOLs performed really well to deplete ribosomal RNAs, leaving only about 1 to 2% remaining rRNA. And what's more, the information from the non-rRNA portion was more rich in terms of transposons and non-coding transcripts. So they could see more of the molecules that they were studying and what was involved and what was taking part in the regeneration process. They also found that the riboPOOLs gave more reproducible results. As you can see here between the triplicates, these were more reproducible compared to the poly-A libraries. So we were quite happy with this result, and we went on therefore to create more riboPOOLs for other species. So we went on to create the human and mouse riboPOOLs, since they're the most well studied, and with help of our colleagues at the University of Würzburg, at the Core Unit Systems Medicine, the sequencing unit, and as well as our neighbors, IMGM laboratories for the RNA sequencing, we were able to validate the riboPOOLs and saw that when compared to Ribo0 from Illumina, these really performed pretty well, depleting about 10% more ribosomal RNA than the Ribo0, both for human as well as mouse riboPOOLs. And reproducibility was also very high between the replicates. So we were quite happy with this result, and since then we've produced a range of different riboPOOLs, and so far we've had good feedback from these riboPOOLs designed against different species, typically depleting greater than 95% of ribosomal RNA. Being hardcore RNA scientists, we also did a northern blot where we looked at the depletion experiment, and we isolated the beads which had the hybridized rRNA and probes, and we checked for the presence of the ribosomal RNA, and we saw that these were only on the beads and not in the supernatant, whereas a control RNA, in this case U6 no RNA, was seen in the supernatant but not on the beads. So it was quite clear that we saw a complete removal of the ribosomal RNA. And we were curious to test how much complexity is required for the biotinylated probes. So we did also a small experiment ourselves to check this. We made different mixtures of 11 oligos each against human 28S rRNA. And you can see we analyzed the depletion efficiency by real-time qPCR. And the graph on the left shows that when using single oligos, so in this case, oligo 3, 6, or 27, there was hardly any depletion. However, when using a mixture of 11 oligos each, you can see there is some depletion, but still the effect is quite variable, with mix 3 not performing as well as mix 1 and 2. And when combining all 3 mixes or even 2 mixes, this depletion efficiency was much better and more reproducible. So this is the zoom in on that. So certainly this pooling also seems to work for biotinylated probe pull downs. So we've made, as I said, a lot of riboPOOLs so far since we started selling them in early 2019. And we have a range of riboPOOLs, not only for samples containing single species, but also samples containing multiple species. We have a diverse range of riboPOOLs, anything from zebrafish to fly to insects, yeast, sponges, as well as clams. And prokaryotes, we also have dedicated riboPOOLs for these prokaryotes, including archaea bacteria. What's interesting, however, is the use of the pan riboPOOLs. When someone wants to study multiple species in a sample, or they want to use one riboPOOL across several samples from different species, we created pan riboPOOLs, which is one riboPOOL designed against conserved rRNA sequences between closely related species. And we have eukaryotic pan riboPOOLs and prokaryotic pan riboPOOLs. And many of them have been successful. We have new riboPOOLs coming up, the pan mammal and pan fungi. And we have the pan prokaryote, which is the most well used so far. We also have something called combination RiboPOOLs, which are basically mixtures of up to 4 existing RiboPOOLs, where it's used for a single-step depletion when the sample contains very diverse species groups. So this can be applied to metatranscriptomics, where you have soil samples, for example, containing plant and bacteria, or even pathogen-infected tissue. You can combine up to 4. We can do the combination for you, and you can purchase this, or you could essentially buy separate RiboPOOLs to combine yourself. And the ratio of combination is determined by the user and is meant to reflect how much of the RNA from each species is present in the sample. We've also made RiboPOOLs against other abundant rRNA, or rather RNA. So human globin mRNA is very abundant in blood cells or blood samples. And we've made that to deplete this mRNA before sequencing. And we also have a poly-A tool which is used for enrichment, but we're still awaiting the sequencing results for that. And if you don't have a species that is listed here, it's very easy to create a custom RiboPOOL with a one-time setup service, following which the RiboPOOL will be available as a standard RiboPOOL. So there's more information about our pan-prokaryote RiboPOOL. It was designed to give a wide coverage of different species of bacteria as well as archaeobacteria. And we've gotten very good feedback from different labs. And here's a list of bacteria that have been validated by RNA-Seq. So depletion efficiency ranged from 65 to 99%. Of course, it really depends on the sample quality as well, how degraded the sample is and so on. But overall, it performed very well in terms of targeting a large number of different species. And we've now also created more specific pan pools against bacteria and pan archaea to narrow down on your studied species. And we've also come up with a new pan plant RiboPOOL, which was designed against a broad spectrum of flowering plants or angiosperms. And this depletes cytoplasmic as well as plastid and mitochondrial RNA, so can be applied to all different kinds of plant tissue. And here we have actually nice data from a customer who has tested this, and I'll go through this briefly here. This was given by Dr. Bruno Hürtel and Dr. Christian Rohler from the Max Planck Genome Center of Cologne, where they used the pan-plant RiboPOOL on different species of plants here, as you can see, from different genera. And you could see that the depletion was very successful, with about 3 to 10% ribosomal RNA remaining. And what was interesting was even though the RiboPOOL was designed against these angiosperms, there was also a distant species of a primitive green algae called the Chlamydomonas, which happened to also be tested. And we saw a good depletion efficiency even of that species. So it's very distantly related, but yet because it shared conserved rRNA sequences with the other species, the RiboPOOL also worked for that. It was not as efficient as the others, but still a very decent depletion efficiency of 80%. So this was the conditions which the experiment was run in. And we also have other customer data with RiboPOOLs. This came from Elio Rossi from Denmark, and he was looking at P. aeruginosa from clinical isolates cultured in LB media, and he was mapping these to P. aeruginosa, but of course the clinical isolate may contain other bacteria, hence you don't see 100% sometimes. But you can see that depletion efficiency was very high for many of these samples, leaving less than 5% ribosomal RNA. In some cases, less, but mostly performing very well. We also have results from our S. cerevisiae RiboPOOL. So they used the cerevisiae riboPOOL in samples containing both S. pombe and S. cerevisiae. And what was interesting was that the riboPOOL not only worked very well for S. cerevisiae, leaving less than 1% of ribosomal RNA, it also worked for S. pombe, leaving 10% ribosomal RNA, because these species, I guess, shed some conserved ribosomal RNA sequences. So as I mentioned, we offer combination riboPOOLs for more complex samples, and these are a mixture of up to 4 riboPOOLs combined at custom ratios that are suitable for metatranscriptomic samples. We've tested it so far. We've tested the human, the mouse, and the pan prokaryote riboPOOL mixture combination on samples, high-quality RNA samples from E. coli, human, and mouse. And by bioanalyzer, we see that with the single species riboPOOLs, you saw efficient depletion of the rRNA peaks. And with the combination riboPOOL, you also saw the removal of the rRNA peaks. And this was seen also by RT-qPCR. And we've heard also back from customers using these combination riboPOOLs with good success in samples containing mixed species. Another advantage of riboPOOLs is that it can be used with a wide input range. So from 100 nanograms up to 5 micrograms, we've seen good depletion success. Here with bioanalyzer, we saw removal of the peaks from 100 nanograms to 5 micrograms. At 100 nanograms, the detection limit is reached by bioanalyzer, but with RT-qPCR, you could still see that the ribosomal RNA was removed, whereas normal or coding RNA was remaining. So the workflow is quite similar to RiboZero, if you have used that before. It's basically coming in a kit, but you can also order them as probes alone if you want to create your own buffers. We give you the recipes and so on. What is involved, as I mentioned, with basically single-stranded DNA probes. This is the riboPOOL, and this is a complex mixture. This is added to the total RNA, and hybridization occurs at temperatures from 68 degrees, and this is left to anneal, after which the streptavidin-coated beads, magnetic beads, are added to the sample, and with a magnet, this can be removed. This will remove the hybridized rRNA and probes, leaving only the relevant RNA, which can be cleaned up by a variety of methods like column purification or bead purification, as well as ethanol precipitation before library preparation. And the time required is around 17 minutes when using a column purification method. So in summary, the riboPOOLs consist of a complex pool of optimally designed oligos, which helps to increase the specificity as well as the efficiency of the depletion of the ribosomal RNA. And on top of that, the riboPOOLs offer multiple benefits. They can be made for any species or abundant RNA. They can be used to detect small or long non-polyadenylated RNAs. They're suitable for complex samples, and they have a broad RNA input range. They can also be used with a fast workflow and very affordable, considering if you don't have a tool for the species you're studying. And they're also HPLC purified, so there's less contaminants, risk of contaminants. We're very happy that we have the full kit as well available since last year. And this is available in 6, if you're using it for the first time, 12, 24, and 98, 96 reaction sizes. They are shipped at room temperature. They come with all the buffers and the beads and ethanol cleanup reagents you need before library prep. And as I said, we also have the probes alone if you want to use that, where we provide you the list of where you can obtain the other materials. And this is available from Biosome Scientific if you're in Germany and Austria. Otherwise, you can also order them through our website. Okay, thank you for your attention. Happy to take questions.
Yeah, okay. So hope you can hear me. Thank you, Catherine, for your great talk. As Catherine mentioned, in Germany and Austria, you can contact Biosime directly for information, quotes, or more questions. In other countries, please contact siTOOLs or your local distributors. So now we will start answering the questions. As mentioned before, just type them into the chat box. And here comes the first question. So let's start. What is the difference between the pan-prokaryotic pool and the pan-bacterial pool?
Right, so let me go to that slide. So pan-prokaryote riboPOOL was the first riboPOOL we made, targeting microbes, bacteria, and it targets also archaea bacteria. So it targets basically all these groups of species. The pan-bacteria riboPOOL is a second generation riboPOOL recently launched, and that targets just the bacteria portion, so this portion. And the reason for doing this is with having a complex pool, you also sort of dilute the efficiency if the species that you're studying is not in that. So if you're looking at only proteobacteria, for example, you're not interested in archaea, then it's better to use a pan-bacteria riboPOOL to kind of have the highest possible efficiency for our RNA depletion. So that's the main difference.
Okay. Oh, here coming up a lot of questions. So next one. Will these kits work for any NGS platform, e.g. Illumina and Ion Torrent?
Yes, so they work for many different platforms. I think from the customer data, it was mostly Illumina sequencing platforms, but I think also with Ion Torrent it should work.
Okay, thanks. Oh, next one. Can I use the RiboPOOLs for degraded RNA, e.g. FFPE?
Yes, I was hoping you would ask that. So I have another slide. It was a bit complicated to show. So this is also from Elio Rossi in Denmark, and he was looking. He was using the combination riboPOOL, so a human and pan-prokaryote mixture of 78:22 ratio. And this was on cystic fibrosis patients' sputum samples. So he was looking at depleting ribosomal RNA from these samples, and these samples contain both human and bacteria, and are also not the best quality. So this is DV200, which is a substitute for the RIN. It basically means the percentage of fragments greater than 200 nucleotides. So the lower it is, basically, the more degraded it is. And you see that even with low or degraded samples, you see a very good efficiency of depletion. And of course, some samples you don't see such a good depletion. So it's very hard to sort of nail down the ratio to use. Initially, he was trying a 10:1 ratio, and that worked for some samples and not for others. But here it worked for more samples. But again, yes, it does take some optimization to get the ratios correct. But we see that overall, there was a good depletion efficiency, leaving about 20% ribosomal RNA remaining, and even for degraded samples. So with the high complexity of the riboPOOLs, this really helps also with the degraded samples. However, we are planning to come up with another new riboPOOL. So this will be for ribosome profiling applications. And in ribosome profiling, you have very fragmented RNA down to 17 to 35 nucleotides long. So we are creating a riboPOOL that with a greater coverage, so no gaps between ribosomal RNA for that. And that should hopefully perform also much better for degraded RNA. So stay tuned for that, for the new develops on that.
Okay, I have another one. How does RiboPOOL perform for RiboSEQ rRNA depletion?
Sorry, I didn't get that.
I think he asked for, do you have data for RNA-Seq data? Because he's asking how does RiboPOOL perform for RiboSEQ
Ribosequencing. So ribosequencing is exactly ribosome profiling. Uh,
Yeah.
and we've had variable results with that. So one customer showed a 70% depletion efficiency, but for other labs, it's less efficient. And we think it's like the sample preparation and it's quite variable between labs. So you can certainly try the RiboPOOLs for that, but we recommend waiting also for the new RiboPOOL, as I mentioned, that will be coming up specifically for this application.
Okay, I have a few other questions about ribosome profiling. So, do you have more material what we can provide to the interested customers?
For ribosome profiling, we have on Select Science, there's a review on RiboPOOLs for that. So if you go to Select Science website and key in RiboPOOLs, you'll have a review from a user describing their feedback on using RiboPOOLs for this application. Yeah, that's but we are working as well on getting this solution. So we'll provide more information once available.
Okay. So I would say, okay, last question. Do you already know a launch date for fungi?
Oh, I think that is so we have Pan plant to be launched very soon, so in July. Already we can start selling them. The next we're thinking is Pan mammal, and I think Pan fungi will come after that. So probably in 2 months' time, 2 to 3 months' time, I hope.
Okay, so at that point, I would say thank you for the questions. We have some questions left open. I think we will answer them in an email later on because there have been a lot of questions here and we limited the time for now. So I would say thank you to all of you. We hope that you see one on another to another webinar from us and have a nice, wonderful summer evening. Bye.
Bye. Thank you.